So, like I stated, my name is Helen Tekla, and I am the education manager here at Cure HHT. Like I said, I'm really excited to have Dr. Marchuk here today presenting on discovering the HHT genes. This is going to be a fireside chat, so we're going to have about 40 to 45 minutes of presentation, and then we're going to have another about 40 minutes of Q&A. So thank you to everyone that submitted the questions during registration process. During the specific webinar, we're going to be focusing on how the three HHT genes were found, what they do in the body, how the mutations cause HHT, and what it all means for the search for better treatment and a cure. So before we jump in, I wanted to make sure that you are all aware of some important news and announcements. If you have not received any emails yet, we are going to be having another exciting webinar coming up soon. When it comes to HHT, early recognition can make all of the difference. Yet too many children remain vulnerable to late or misdiagnosis. In this educational session, Lynn Soscarsi, will discuss how to recognize symptoms of HHT in children, understand diagnostic criteria, and navigate care management, referrals, and treatment options. So please join us. This is going to be Friday, April 10th from 12 to 1 p.m. She is the HHT Nurse Program Coordinator at the HHT Center at Sisters of St. Mary Health in Children's Hospital that's located in St. Louis. Lynn is a longtime advocate for pediatric patients and families, and she brings more than 25 years of experience to this important conversation. So registration is now open. We will be dropping the link in the chat, or if you cannot see that, please head over to our website for more information, and we hope to see you there. I also wanted to remind you of two ongoing programs here at Cure HHT, both that are aimed to help the trajectory of this disease. The first is HHT Connect. It is the first ever global HHT patient registry. Our goal is simple, and that is to collect health information from as many HHT patients as possible so that researchers and doctors can discover patterns and clues that can help them better understand HHT and develop new treatments, which is exciting. By answering a handful of simple questions, you can help advance HHT research, all from the comfort of your own home. And I would strongly suggest everyone to utilize the link in the chat and visit our website to learn more and sign up. Our next program is CHORUS, and that's the Comprehensive HHT Outcomes Registry of the United States. It's an observational registry of patients diagnosed with HHT. The purpose of the study is to better understand HHT, the symptoms and the complications it causes, and the impact that the disease has on people's lives. The investigators will collect long-term information about the participants, allowing us to understand how the disease changes over time and what factors can influence those changes. And ultimately, this should help improve treatments for the disease. Right now, many HHT centers in various regions of the United States are recruiting patients to participate in the study. If you'd like to learn more, please head over to our website, but you can also email chorus@curehht.org. There will also be a link in the chat that you can access. And if you want to learn more, if you want to sign up, or even if you have any questions, please do not hesitate to reach out to us. And finally, before we get underway, I did want to go over some housekeeping rules for the webinars to make this event as interactive as possible. One, I'd like to just thank everybody who submitted their questions during the registration process. We got a lot of great questions. So at the end is when we'll be hosting a moderator Q&A session. This will be to we recommend that you do keep your questions in general so that they benefit everyone. And then just for everyone is aware that the presentation is being recorded and it will be shared in the Cure HHT website resource library. So please look out for that email with the on-demand link within the next week. So with that being said, it is my pleasure to pass it off to Marianne to introduce our speaker for today, Dr. Marchuk.
Hello, everyone. It's really a privilege for me personally and professionally to introduce Dr. Marchuk. I have known him for nearly 30 years when I was president of the board of directors. And he is the James B. Duke Distinguished Professor of Molecular Genetics at Duke University School of Medicine. He was born and raised in Cleveland, Ohio, and earned his Bachelor of Science degree from the University of Dayton. He went on to complete his Master of Science at the University of Connecticut and received his PhD from the University of Chicago. After conducting postdoctoral research at the University of Michigan, he began his work on HHT in 1992, embarking on a search for the genes that, when mutated, cause HHT. For more than 3 decades, he has continued to perform research on the underlying molecular and genetic events that cause HHT and has a dramatic impact on our science and understanding. Please help me in welcoming Dr. Marchuk.
Should I go? All right. Well, first off, thank you, Marianne and Helen and Cassie, for this opportunity. I want to say hello to everyone and say good morning or good afternoon or good evening because I think we've got people from all over. I'm going to, this is really a privilege for me. In a sense, I'm not actually meeting you in person, so I don't see you, but I'm really been thinking a lot about this presentation and I want to give you the best shot at what I hope to do, which is to tell you about HHT, how we discovered the genes, and a little bit about, as the subtitle says, the journey from genes to mutations to therapy. And I want to start out by saying a welcome from my institution, Duke University. It's an absolutely beautiful main campus, and it's nestled in the woods here with this beautiful chapel. Some say this is the second most famous building on campus because, unfortunately, the first most famous is Cameron Indoor Stadium, where the basketball team plays their home games. They're playing this afternoon in the NCAA tournament as the #1 seed, and that's a harder ticket to get into the stadium. The campus is really beautiful, and I want to just show you. This was taken last spring. These pictures just along the campus. You see the beautiful river stone they used to create the Gothic um, architecture of these are classrooms and dorms. It's really beautiful. And the medical center looks a little more modern. It's sort of adjacent to that, but it's a beautiful medical center and a really important one. And here at this point, normally when I talk, I say, anybody nearby coming this way, come, we'll have lunch. We'll take a tour of the lab. We'll show you some things. But you have to do that before June 30th because I'm retiring on June 30th and for a lot of reasons, but that'll be the last time I can bring you into the buildings. And one sort of caveat here is this idea that what we're doing here is trying to educate you, give you information, but we're not giving you medical advice. And this is neither myself nor the foundation is trying to give you medical advice. And you really need to consult your healthcare providers, especially at a Center of Excellence, which I'll talk about in a little bit later. So we're going to start with a short primer on human genetics and genetics in general. And this may be elementary to some of you, but we want to start where everyone's the same place. So first off, everyone knows, I think, that HHT is inherited as an autosomal dominant trait. And that means that inheritance of only one mutant copy can cause the disease. And it could come from the mom or the dad. And for each child, there's a 50% chance of receiving a faulty copy of the gene. And so therefore, sons and both daughters and sons can be at risk. And by the way, this doesn't mean that if you had 4 children, 2 would have HHT and 2 didn't. It's just like flipping a coin. Every child is a new coin flip. So what is being inherited? Well, the DNA is encapsulated in the nucleus inside of every cell in your body. And there's 23 pairs of chromosomes in the cell, pairs because you get one from each parent during growth and birth. There are 22 what are called autosomes, so these are the non-sex chromosomes, and then you have the sex chromosomes. Since HHT is inherited as an autosomal dominant trait, you know that it's inherited on one of these autosomes. And what are those chromosomes made of? Well, they're made up of DNA, the double helix, deoxyribonucleic acid, And each chromosome is basically highly compacted DNA. So here we see the famous double helix in cartoon form, and you're going back and seeing at different levels of resolution. The DNA is wrapped around proteins called histone. These are wrapped around in nucleosomes, solenoid, and then this DNA is really compacted so that a chromosome is this spaghetti-like thing that's super compact and billions of base pairs of DNA all in every cell. And it's the sequence of the letters, which we call the nucleotide or chemical bases, that determines the code for the gene. So, how many genes are there? Well, there used to be thought to be hundreds of thousand genes, but now it's the current estimate is approximately 25,000 genes. But nearly every gene has multiple sections that are spliced together. We'll talk about that when we talk about mutations. And that splicing pattern, if you know, A, B, C, D, you can skip exon B or exon Z, so that the point being is that you could make many more combinations for each gene of transcripts and proteins. So every gene may encode for multiple slightly different versions of the same protein. So every cell has the same amount of DNA. Well, then how do we get something like a brain cell or a pancreas or a bone cell, or for HHT, an endothelial cell, the cells lining the blood vessel? So if all the cells have the same information, what happens? How do they function differently? And the idea is, and this is called the central dogma of molecular biology, is the DNA is copied into a messenger RNA form, and that messenger RNA is an exact copy of that of the gene, and and that is called expression. So the genes are expressed, and each cell is expressing a different set of genes, making a messenger RNA for a different set of genes that ultimately leads to them having different shape and function in the cell in the body. So that and then that messenger RNA encodes that same DNA information, and it is copied into a chain of amino acids that eventually, as they're put together, fold in a way that forms a protein. So each type of cell, whether it's a bone or a blood vessel or brain, is instructed to make a different set of proteins. And that's through this messenger RNA. So then, well, then how does a mutation cause an inherited disease? And you could think about it in a simple way, in this almost cartoon version, that when there's a mutation in the DNA and that's copied, that messenger RNA has that mutation code, and that causes a different chain of amino acid to be put in that chain. And so the protein is different. And if it's severely altered, it could cause disease. So the faulty protein in this case wouldn't perform its normal task. So let's talk a little bit more about the genetic code. And I don't want to get into too many details here, but this is one way of showing the genetic code. And it's like a wheel. And there are 20 amino acids. Sometimes they have one letter code, sometimes they have three-letter codes. These are the 20 amino acids around the outside that constitute proteins. But the code is such that it's a triplet. So the DNA code is a triplet, and whoa, T, or C tells the ribosome to put phenylalanine in the growing protein. T, A, or G, we're reading from inside outside, says to put leucine. T, C, anything says serine. So you can see there's some redundancy here. So most but not all DNA mutations will alter the amino acid, but if it went from TCT to TCG, it wouldn't matter. It would still be serine. There's one important point because we're going to talk about mutations. There's no punctuation in DNA. So there's no spacing in between the letters. There's no periods. There's kind of a period with a stop codon in the sense that these three, TAG, tell the protein chain to stop. And then they have these historic names, ochre, amber, and UGA never got a name, but okay, so that's the code. So that code is read by the ribosome in the messenger RNA that's just a direct copy of the DNA, and then the proteins are made. So we're going to talk now. Okay, so HHT mutations are found um, um, some DNA changes are easily categorized as mutations. And here I'm using the word mutation not to say it's a change in the DNA, but it's a change that causes the disease. So I'm going to call it a disease-causing mutation. And so I've borrowed some of these slides from the National Human Genome Research Institute. So here we have the messenger RNA. Notice that there's a bunch of codons. This is the three letters, the nucleotides going in threes, and they encode for a different protein like methionine, threonine, aspartic acid, glutamine, protamine, proline, etc. Now, what if we put one of those stop codons? We gave the instruction here UAG, that's a stop. And so the protein would stop being made. And if it was supposed to be going on dot 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 to this big long protein with 200 or 300 amino acids or whatever, and it only got stopped at 50, it's going to make this dinky little thing, and that is not going to have a good function. So that's called a nonsense mutation. It's nonsense. The protein essentially is complete nonsense. It just doesn't work. And that's an easy one because there's only three codons that say stop, and if it shows up prematurely, it's an easy mutation for the diagnostic lab. Say, that's a mutation. We agree. Another one is called a frame shift because remember I said that the code, the three-letter code, has no punctuation per se, and it definitely doesn't have any spacing between the words. So we've got all this going on, three, then you read the next three, then the next three. What if the DNA replication inserted an extra base, an extra nucleotide? All of a sudden, from that point on, you're still reading three, but you're now out of frame. So now it's shifted, and we're making not the same, we're making the same, we're incorporating the same amino acids up to that point, and from then on, we're out of register, we're out of frame, and invariably there's a stop codon happens soon because now you're out of the right reading frame. So whereas you might have had a beautiful, again, full-length protein that was folded correctly, here you've got this protein. At some point, it was correct. Then it frame shifted because of the insertion, or it could be a deletion of a base too. You'd be out of register -1 instead of +1. And then the wrong amino acids were added because now you're out of register. And then invariably you have a stop. This is junk too. So these frame shift and nonsense mutations, they're really easy to say, I'm calling that a mutation. There are some that are more difficult, and I'm going to show you this here. This is called a missense mutation. Well, this is what we, a priori first think about when we say, oh, we made a mutation, is the sense that we changed one of those nucleotides in the code, and instead of encoding tyrosine, it put in serine. Everything else is the same. It's just that one word, one three-letter word, the codon, was wrong because one of those bases was a mutation. So then you get a full-length protein, and it has one little place that's wrong, that's incorrect. But often those will fold correctly. Sometimes they'll fold incorrectly. It's just hard to predict what that does. So let's think about this more. How do we interpret? We can find the missense change, and we know how to interpret it to say that it's a tyrosine to a serine. How do we know that this protein, we know this works, this is the normal copy, the normal version. How do we know that this won't fulfill its function? Okay. So here in first line, this is too much words, but I wanted to point this out. Let's use endoglin as an example. It has 658 amino acids. So each position is encoded by one of a codon, those three-letter nucleotides. And each one of those 658 positions has one of 20 different amino acids. But a mutation in any one of those could show a missense variant. So all 658 are liable to have a DNA mutation in them. And then we have to interpret what that means. But how do we know what's going to happen? So what if, I think this is an overestimate, but math is easy when you multiply by 10. Let's say, based on that codon wheel, every codon can change to up to 10 amino acid difference, 10 different amino acids. So that's 658 times 10. That's 6,580 different possible mutant proteins. Now, no research lab can synthesize all those different possible versions a priori ahead of time and test them all out so they can tell the diagnostic lab, these are mutations because they impair endocrine function. These are silent. They're benign, they say. They don't have any functional consequences. That's just too much work to do in the lab. It just can't be done. It can't be scaled. And so, more importantly, even if you say, well, you don't have to do all of them, let's just check the ones that we find in patients and see whether they're functional. And that's would work. But a diagnostic DNA testing lab is not equipped to do functional studies. That may take a few weeks, a month, maybe more, of some dedicated person in the lab synthesizing that protein, doing functional studies, putting it in cells. That's not what they do. They're doing straightforward tests where they run lots of tests and they turn the crank. So the point is, missense mutations are harder to distinguish as mutations versus just benign changes. Now, one way you can tell, we're going to talk about this in a minute, if multiple times different families keep showing the same change, that sounds like that's going to be a mutation. There's other things we look at. And so someone I saw in some of the questions, how about AI? Well, there are computer algorithms that will predict the effect of any change. So I said there were 6,500 possible changes if you do the math, probably a little less than that. But the point is they do, in fact, those programs predict what if position number 343, which is a serine, becomes a leucine or an arginine or whatever. And then they will go through the whole thing computer-wise, and they're using evolutionary conservation data, like what do other species look like for endoglin, for example. And then they're using understanding of what the amino acid chain side chains look like and all sorts of other data, and then they predict. But these are predictions. They're not fail-safe. And so diagnostic labs, they always use these. And with other evidence, it'll tip it over the edge. But this alone is not, using these computer algorithms alone isn't often enough to categorize a sequence change as a disease-causing mutation. They found the missense change. Is it really causing HHT? Okay, another tool, more too many words, but another tool is looking at the ever-growing database of DNA sequence in large populations. Here's the idea. If a sequence change is present in the normal population, especially at some modest frequency, well, then it probably doesn't cause HHT. By contrast, if you never see it in normal people, and we're talking about hundreds of thousands of people, maybe a million people, maybe more, these databases are growing, just sequencing people. Like you've heard of the NIH, all of us. They're sequencing all sorts of people. They want to get up to a million people. If it's completely absent, then in normal people, and you find it in a family, well, it's probably a disease causing. But the assumption here is these large databases do not contain HHT patients. And I'm glad Helen asked that question at the beginning because most of you had a diagnostic odyssey where you had HHT and you had the symptoms, and it took a long time for someone to finally diagnose. So when they go and start sequencing random people in the population, some of those people might have HHT and they never knew it. And they're in that database. And furthermore, these databases are anonymized. It is a word. I looked it up. In other words, we don't have it, I can look at all these databases at any time, but they don't have the clinical information. They don't have the patient information. I can't get back to them. So that's for patient privacy. Great. But then, so we see something in the database in normal people. Is everybody in there normal? So if you see it only once, that patient might have had HHT. So you can't always use these population database. All right. Here's where I think one of the real interesting things about identifying and understanding mutations are splicing mutations. And this is a cartoon version of a gene with only 3 exons. The endoglin and ALK and SMAD4 have many more exons. But the idea is in the DNA in our cells, we have little regions of sections called exons that have the information for making the protein. And then there's this intervening sequence, which is just not used, and it's spliced out at the messenger RNA level. So the messenger RNA is made. It's a huge transcript with these black sections are what's going to encode the protein. And these parts are spliced out so that the mature messenger RNA that the ribosome sees has all this spliced out. So you can think of this as building building the genetic information from little smaller subsets of genes, smaller subsets of that encode parts of the protein. Okay, so this splicing process is really convoluted and complicated. We know some of the rules. There's a at the beginning of one of those pieces of DNA that's going to be it's going to be at the messenger RNA level. It's going to be spliced out. There's an AG, and then the next exon starts. There's a GT at the back end of this. But I want to show you this. What does this mean? It means there's a G or a T, but then there's an A or a G, and then usually an AGT. But I want you to look at these data down here. Let's look at right here. I want to show you this one right here. I think this is this one. Yeah. 100% of the time, this is A and this is G. So if there's a mutation there, it's not even in the region that encodes the protein, but it's in the information that says splice these two sections together in the messenger RNA. I want you to look at the next nucleotide here. 70% of the time, it's a C. Well, 24% of the time it's a T, 4.2%. You see, there's redundancy here. Look at the next one. It looks like it could be anything and it's random, but it's not random. So any of these nucleotides could affect the splicing pattern or maybe not. And so one has to do some more testing at the messenger RNA to see if there was a missplicing, if an exon was removed and the next exon, you went from exons 1 to 3 and skipped number 2, or maybe you skipped 2 and 3 or whatever. So no diagnostic lab can do these messenger RNA studies for every putative mutation they find. I'm using the word putative to mean they found something. They think it might affect something, it might affect splicing. It's not even in the coding region, but yeah, but they have to do an experiment and they won't do an experiment. Okay, so here's the take-home lesson. When a diagnostic lab can't interpret the DNA sequence change that they found, even when it's in one of the known HHT genes, it's surrounding in the intronic region, the spliced out region, or in the coding region, or just up and down where there's regulatory elements that turn the gene on and off. If they can't be sure that they can interpret how it works, they're going to call it a VUS, and that stands for variant of unknown significance. And it's a little bit, maybe you say punting, they're going to, I don't, throwing up their hands, but they just can't follow every one of these. Okay. So the take-home lesson here is there's so many different ways to break an HHT gene, or any gene for that matter, and their proteins. And not all the easy mutations are easy to find or interpret. Thus, many genetic tests will result in the VUS, which is really dissatisfying for you. I want to talk about one more kind of mutation that's not easy to find. If there's a big deletion, if in other words, the DNA is gone on the copy that you got inherited from the parent with the disease, so the disease is gone, the whole gene is gone, or a big part of it, then when you're sequencing, all you're sequencing is the normal copy, and then everything looks normal. So there's another example. Deletions can be identified, but they're not as easy to identify, and they take a little extra work. So I want you to think about this for the implications for therapy by mutation correction. I just said there's so many ways to break the gene, and nearly every HHT family has a different mutation. So if you think about it, correcting the gene, trying to correct that exact change, is going to require a unique therapy for each mutation in each family. And therein lies the difficulty of trying to do gene correction for a disease that's called a loss of function disease. That is, the mutations cause the protein to lose its function. There's so many possible mutations, and they're all over the place. Some of them maybe even not even understood or identified correctly. So it's going to be really hard to do gene correction. But perhaps a better strategy is to add back a normal copy. You say, look, this person has some weird endocrine mutation or a weird ALK1 mutation, ACVRL1. Let's just add back a normal copy and not worry about trying to correct a different mutation for each family. And these so-called gene replacement rather than correction therapies make a lot of sense for HHT. There's still some caveats, and I have some data here. You've got to deliver this gene therapy in some kind of way to deliver it to every endothelial cell. You'd be surprised. Yeah, we should take a little survey. How many endothelial cells do you think are in an adult human? And not that every cell will be form of vascular malformation, an AVM or telangiectasia, but you don't know which you're going to do it. So you probably want to not correct, but you probably want to deliver the gene back, the correct gene to every cell in the body. Well, it's hard to know exactly, but the estimates are between 1 and 60 trillion, trillion, not billion, not million, trillion endothelial cells in your body. And you might have to correct them all, or at least localize the correction to certain organs and organ systems. So that's the point being, I think, is that a gene replacement therapy, I think, is really still viable for HHT. And a mutation correction would be viable, but you can't do a different therapy for every single mutation. It'd just be not cost effective and no one would design it. So that was my primer on human genetics, and I tried to anticipate as Helen sent me some of your questions that you'd sent in earlier. So try to give some information. We can still talk about those questions, but I wanted to have a little bit of information in slide form, excuse me, so that you could see it and we could talk about it in a better way. Just excuse me. Okay, so how did we find the HHT genes? Remember that we took a blood sample and got DNA out of your white blood cells. There's 3.3 billion base pairs. How did we find the mutation? How did we find even which gene was mutated? And I think there's a couple of things I want to point out. The first thing we needed was what we call genetic maps. And back in the early 90s, they were I'll show you what I mean by a genetic map in a minute, but that was courtesy of the Human Genome Project. So before the genome was sequenced, we had signposts, maps, like little regions of the genome where we knew this is next, then very far away is this, and then this, and each chromosome had a map. And then we needed to collect DNA from large multi-generational families with HHT. Multi-generational, so we could see the disease passing through the generations, and we could correlate that with pieces of DNA and which one seemed to go, which region of the genome, which chromosome, which region in the chromosome seemed to correlate with the people getting the disease. So here's a little cartoon version. Here's the first genetic map, but that's not very good. It's just a bunch of chromosomes and we're looking at them. We don't have any real detail of what's going on that chromosome number 5, for example. So this is called a karyotype. Everything is there, but there's no resolution. So where are the genes and which genes were mutated? So we had to use a map. And the first stage of the Human Genome Project was to create genetic maps. Here's what I mean by a map. This doesn't look like very helpful, but what we're saying is we know there's a piece of DNA here, and it's in recombination units, don't worry. And then it was at position 14.7. And very far away, we had this. It's almost like the state of North Carolina is pretty long, and it's almost like knowing a house in every county or two where you had the house. And you don't have anything in between, but you know about the house, and then the next county you have a house in a little town, the next you have a house in a larger city. And so all these positions, we had maps. And we started to look at all 22 pairs of autosomes. We didn't look at the sex chromosomes because it wasn't inherited in males or all females. It wasn't sex linked. And we started looking at every chromosome and just going through and looking which regions of the genome segregated with the disease going in families. And that required here it is. These crude maps gave us the signposts of each chromosome. So the other thing we did was we collected large families. And by large families, I mean families that are so large. Now, we know of people in this first generation here. We know their names from family history. Nobody knows if they even had HHT because it's so long ago. But you can see these are many generational families. The way this works is, if you've ever looked at a pedigree, is the squares are males, the circles are females, and we usually fill in the people who have met the diagnostic criteria for the disease, and the people who didn't are open circles or open squares. And so we have blood on as many of these people as we could. And these were our first two families where we located the first gene. We didn't know what the gene was, but we knew it was on chromosome 9, and not just chromosome 9, but a position near the end of the chromosome. And the reason we did the way we did that was we had signposts all along all the chromosomes, and a piece of DNA somewhere near the end of chromosome 9, it was inherited always is a variation in that sequence on chromosome 9 was inherited every time a person got the disease, and they didn't get that variant of that piece of DNA when they didn't get the disease. By the way, these two large families were collected mostly before I even started. One was, and I'm losing track of which it was. I think this is the one that was collected by Alan Guttmacher in Vermont. And I toward the end of the collection process, I flew out there there's some French Canadian blood in here, and we went up to the border of Canada, and we were literally looking at gravestones, trying to put the pedigree together to see who was related to who and who married who by looking at gravestones in a French Canadian graveyard. This family was collected by Mike Iannuzzi and Francis Collins of the University of Michigan. And so Marianne alluded to this, but when I left Dr. Collins' lab as a postdoc, he gave me a gift, the best gift I've ever had. He was talking to Alan Guttmacher, Dr. Guttmacher in Vermont, and he and Mike Iannuzzi. Dr. Iannuzzi was a pulmonologist. Dr. Collins was a geneticist, and they had the box of DNA with most of these people. We collected more afterwards, but they gave me that box and they said, go find the HHT genes. At that time, they said, go find the gene because we didn't know there was more than one. So this is how we found it. So eventually, here's another idea of these signposts along each of the chromosomes. Notice that they're not all, the distance between the signposts is different depending on the chromosome because it was a little haphazard. But eventually, we saw that some families were on this far end of chromosome 9, and we cleverly called that HHT1. And then other families were on chromosome 12, kind of more in the middle. That was called HHT2. Remember, we don't know the gene yet, but we know that some families have something here, some families have something here. And much later, in a process that I won't get into, where juvenile polyposis was already known to be here and the gene was known, we figured out that those patients had a combined syndrome of juvenile polyposis and HHT, the so-called JPHHT syndrome. So that's how we did it. And eventually, over the course of the next few years, we identified all three of the genes. And there's the endoglin, there's the ALK, and there's the SMAD. And the SMAD4 is the juvenile polyposis HHT. So, okay, we talked about the fact that these genes are copied into messenger RNA and they make a protein. What do they do? And this is from a review by Jamie McDonald and colleagues. I'm looking at the time. Excuse me, I'm going too slow. But these genes encode proteins on the endothelial cells, the cells that line all your blood vessels. All your blood vessels have endothelial cells. And endoglin is on the surface of the cell, and ALK1 is on the surface of the cell. SMAD4 is inside the cell, and it receives signals from proteins on circulating proteins in the circulation that bind these. They do some phosphorylation events, some signaling events, and these go into the nucleus and influence gene expression. So it was intriguing that all three genes are kind of in the same biochemical signaling pathway, which makes sense because they would have overlapping phenotypes. But here's where I want to be careful, but I want to point out that as the database has grown and more patients have been very carefully clinically evaluated at HHT Centers of Excellence and elsewhere, we've learned now that endoglin possibly has an earlier epistaxis, nosebleeds, a higher pulmonary AVM, arteriovenous malformations prevalence, more cerebral AVMs, HHT2, which is the ACVRL1 gene, later perhaps more hepatic AVMs, more GI bleeding. And SMAD4 was unusual because it was known to already cause juvenile polyposis, and it's just JP plus HHT. But the key here is, I think I've got it here, is this cancer risk because they've already got these polyps in the colon. So that there's people need to be seen both by an HHT center, but by someone who's interested in, colon cancer. And Claire Chauvelin showed that some of these truncating mutations that cause not missense, but frame shift or stop codon, they seem to be associated with more severe phenotype of anemia. And this is what I wanted to point out, though. This is hard, though, because if you look at any given family, oh, everyone's going to get a PAVM because they're in this family. No. So the intrafamilial, within the family, all having the same mutation, the variability can be substantial. So it's not easy to predict. So I was trying to avoid answering questions like, I've got an alpha mutation and I got of this, and my son has this. What am I going to get a cerebral AVM? I think this is very, very difficult to say. And really, people need to go to the Center of Excellence and be screened and discuss how often you need to be screened for based on your family, but also a lot of other information. I had all sorts of stuff about this, and I'm taking up time, so I'm not going to talk about the fact that the somatic mutations. Um, but I want to, because I want to have time for questions, and I think I've gone too long. How did we figure out all this and then eventually find the genes? And what we also found was that in the vascular malformation itself, there are somatic mutations that mutate the normal copy of that gene. Remember, you've inherited one normal copy and one mutant copy from the parent who had the disease. We found that that normal copy then also gets mutated so that that cell has both copies mutated. But the way we did this was by a tissue specimen biobank, and even before that by collecting blood. And the Cure HHT Foundation is really proactive in now and really pushing this tissue bank forward. And the reality is this is going to be good for the future because we really need to know what's going on in those vascular malformations. And that biobank has a lot of issues used for R&D, but you've got consent, but the patients have to be engaged and there's got to be privacy, et cetera. So, and I know Cassie's really worked hard on this, Cassie Friday, who's here to do that. And I just want to end before we take questions to talk about my lab in Cure HHT. I went to the very first HHT meeting in the early 90s. In fact, I went to the first one before it was technically called a HHT meeting in Columbus, Ohio. And then I think we had the first official one in Edinburgh, Scotland. And so I've been at all those meetings. I've missed a few since then with birth of children and other issues. But Cure HHT has participated in almost all my research, really all of it. And it's resulted in some of these significant discoveries because we partnered in over 30 papers, including some that really found the genes and found this idea of the second hit, the somatic mutation. And they facilitated the patient contacts and access to medical information that we didn't always have because none of these patients are my patients. And we've gotten surgical tissue and blood to do DNA studies. It was just invaluable. And importantly, having that collaboration, that association with the foundation, really allowed us to get multiple federal grants, research grants, so that we could, whatever limited money we had, we leveraged that. So my first NIH grant was on HHT, and that was back in 94, I think, 93 actually. And then for 15 years, a group of us had something called the Brain Vascular Malformations Consortium. We had 15 years of continuous funding from the NIH to really study HHT and compare it to other vascular malformations. Wouldn't have been done without Cure HHT. We would have absolutely fell on our face. And we also got a Department of Defense grant, which is kind of funny why it's the Department of Defense. And Marianne someday will tell you that story. But we got that in large part through working with Cure HHT. So we really do need to work together. It's not just the doctors, but also the scientists and the patients and the families. And we will share the insight and information and hope if we work together. I just want to acknowledge the fact that I just told you an odyssey. You've had your diagnostic odyssey. I've had a career odyssey of trying to figure out what causes HHT. And we're going to talk a little bit about other therapies maybe when we get into the Q&A. But these are the people who primarily worked on HHT in my lab over the 30-year period. And, some of these people you may recognize from some of the people on the foundation recognize. I had a couple of people who I need to shout out. I've had a long time collaborations with Marie Fonin, who's the center director in Toronto, Canada. And Cassie, especially more recently, and Marianne have been instrumental in us getting our work done and doing this. But ultimately, it was the patients and their families that allowed us to do this. And of course, money. And there's a lot of information there, but all those different funding sources over 30 years. So I'll end the show and I will even stop sharing. And I think we will go to Q&A.
Thank you so much, Dr. Marchuk. That was an amazing, amazing presentation. Thank you to everyone who's so engaged in the chat and just listening closely to everything that Dr. Marchuk had to present. Um, as much as I would love to keep this webinar going for 2 or 3 more hours, we're going to go ahead and go into our questions. Thank you all to everyone who submitted some of these really, really great questions. So Dr. Marchuk, just to start off with, when it comes to new discoveries, are researchers continuing to discover new genes associated with HHT? For patients whose genetic testing is negative or inconclusive, could this indicate a gene that has not yet been identified or is harder
Yes.
to detect?
So I have, as I mentioned to some of you before we started the webinar, I have very strong feelings on this. And I want to start talking about this in the context of all of genetic disease. So what have we learned from like hundreds and hundreds, thousands really, of genetic diseases where the genes are known and people sequence and try to find mutations? So, and you know what happens? You never find every mutation, especially when you go through the diagnostic labs, because they have difficulty interpreting and they don't really look deep in those introns where there could be splicing errors and stuff that you don't know. So in one sense, HHT looks exactly like every other autosomal dominant disease in terms of its success rate of finding mutations. So that tells us one of two things. Either everybody's missing lots of genes for every disease, or it's really about not being able, with simple DNA testing, to find all of the mutations.
And
And a
how
second
are,
point,
oh,
oh,
yes, go,
so
sorry.
a second point is when laboratories have partnered, diagnostic laboratories have partnered with a research lab, and the research lab starts really digging deep on those patients and families where they could find a mutation, invariably with a lot of work and a lot of not just finding a weird mutation that wasn't sequenced, but then proving that it causes of misfunctional protein or missplicing or regulation. That takes sometimes months of work. But then they start to find these other mutations. So the answer, in my view, is the evidence that there's another gene can't be we can't find every mutation. When I got a negative test back, I must have another mutation, another gene. I think what it means is they've got a mutation that is hard to find. The diagnostic lab won't look for it. A research lab is probably, we talked about this just before everyone got on. The research lab is probably going to be more interested. I'm certainly more interested in not finding every possible mutation, but finding out what's going wrong in that vascular malformation and how can we treat it. That's my goal. Because even my goal is, even if you don't know that you have an endocrine or ALK, if we have a treatment, we'll have a treatment for you. And let's focus on that. So again, the evidence that there's another gene, I think, is unlikely. Doesn't, however, I want to point out something. It is impossible in science to prove a negative insertion. I always say this, if you want me to prove that there's gold in Nebraska, I don't think there is, but if I found a gold nugget in Nebraska, I've proven it. If you say, Doug, prove that there's no gold in Nebraska, I'd have to dig up every square inch in Nebraska. So here's the point. I can't ever say that there's not another gene, but the evidence isn't really there because it looks just like everything else. And it might be that there's some this HHT-like, not meeting HHT diagnostic criteria, but maybe it's they have a telangiectasia or something. So it could be something like that. And those might have different genes, but garden variety HHT, very few people, I think, are looking for it in a serious way.
Well, we got to find that gold in Nebraska then.
Yeah.
So what approaches or technologies are helping move this research forward? Like what are scientists, how are they working to identify these additional genes that have not yet been
Yeah.
discovered?
Well, that's the thing, right? The technologies, I think the technologies to find the missing mutations, we'll call them hidden mutations, are there now. It's just a matter of spending the money. So there's something called long read sequencing where you don't read little snippets of sequence so that now if something's missing and you're reading a really big thing, you can find it. If there's a big deletion, you can find that deletion and you could find these weird mutations in parts that aren't part of the coding of the gene. So there's long read sequencing, there's RNA sequencing, full-length RNA sequencing. These approaches, when they're used, they end up finding the great majority of the mutations that were hidden or missing. The other thing is, though, is we've now had really over 25 years of looking for new HHT genes. And, there's been a big goose egg. So other than HHT like and this, so I think we don't need new technologies if there are missing genes. We just, there's going to be one day I'm going to be proven wrong. There's this rare family with this weird mutation, weird gene that causes it only in that family. But that doesn't make sense to me. So it's really the technologies to find the hidden or missing mutations. And they're there now. It's just that no diagnostic lab's going to spend a couple extra $1,000 or more and, weeks or months worth of work of dedicating a person to figure it out.
Is why funding is so important. So are there newly identified mutations within known HHT genes like ACVRL1, ENG, or others?
Yeah, so the idea is that mostly every family has a unique mutation. I think there's going to be a question later about, mutations in isolated populations that spread out. That's called a founder effect. But yeah, so new mutations are found every day, and I mean, almost every day, and that's the conundrum is you're always finding something new. Let's contrast that with sickle cell anemia. You know how many mutations there are in the world? One, actually, kind of two, but one. But the point is, it's either that or nothing. But for here, you've got to sequence the whole gene and everything. There's new mutations every day, but they're all in the same genes. Is the point.
Thank you. How do the different HHT gene variants influence symptoms and complications such as lung, liver, or brain AVMs?
So there's two parts to that question. I showed you how endoglin has it's an overlapping, but a slightly more prevalence of pulmonary AVMs and ACVRL1, the hepatic. Of course, SMAD is very different because it also causes the colon issue. But if you're asking the question, why is that? How does that work? That's been a little bit of a mystery, and I have some thoughts on that that I've shared at meetings. But the bottom line, and it has a little bit to do with the fact that if these vascular malformations are seeded by a second somatic mutation, then that's going to be a little stochastic and random. But why brain is more prevalent in one than the other in pulmonary? It probably has to do with the role of those proteins in that vascular bed in that organ. So the lung is not a brain. And so the lung may have a different need for endoglin versus ALK1 than in the brain. You see what I'm saying? But that's been a long-standing issue in HHT, and we really don't understand why there's organ predilections.
Lastly, from my end, how often do new mutations occur in families with HHT? Can a child develop a mutation even if there is no prior family history?
Yeah, right. So that's called a de novo mutation. If you think about it, every family that we found, if we could go back in time, which we can't, there's got to be a first. We may go back 10 generations, 20, 3 or 4, and that was the first person to get it. Because remember, replication errors, as the DNA is replicating in the sperm and the egg, they will happen. And sometimes they'll happen and cause cystic fibrosis or Duchenne muscular dystrophy, sickle cell anemia. They can cause HHT. So how often do they happen in real time where we've caught it in that generation? That's going to be a little bit rarer, and I don't know the exact frequency. We've seen that in other diseases that I study, but we've focused on these large families, so we've never been able to go back far enough and find it. So it does happen, though, and it can happen. And it's not unusual or strange. It's just you're just capturing it in a snapshot in time. That's the first one. And if we would go 1,000 years in the future or a couple hundred, we'd say, oh, that's a beautiful large family, but somebody had to start it.
Thank you so much, Dr. Marchuk. Marianne, I'll go ahead and pass it on to you.
Thanks so much. And thank you so much for such a great presentation in history. So I have several questions. If someone has HHT symptoms, but their genetic test comes back negative, which many times happens, how should that result be interpreted, and what are the next steps for diagnosis
Yeah,
for this family testing?
I think the one thing you said is, let's say they really meet diagnostic criteria for HHT
Right.
and another first degree relative or another relative really meets diagnostic criteria. Then they have HHT. The downside is you don't have the mutation to help guide, um, screening in the next generation of the young kids. But, and so that's the negative, but you don't need to have how should I put this? HHT can be diagnosed and has been diagnosed for years before anybody discovered the genes. So that's the answer. It's just unfortunate that then, because to me, the issue is it's that next generation who, if you had the test, you could say, this child doesn't need to be screened. And we want to be really careful with this child because they've got the mutation, and we want to follow them up pretty carefully because they know they've got it and you don't have that advantage. And unfortunately, like I said, it's just a matter of no one's going to go to the heroic efforts to find every mutation. It's just too
Right.
expensive.
So you touched upon de novo mutations. What does it mean with a de novo or being mosaic
Yeah,
for?
so the de novo is you being the first person. So you
Right.
shouldn't think, oh, my parents didn't have it. And frankly, you have to be careful here. And you may know where I'm going with this because and we've actually, I shouldn't say this. We've had some cases where mom and dad said, "Oh, look, and when we did the DNA testing, we said, "Oh, mom was usually mom, but it's not always the case that dad, the person who they say was dad." So that's
Right.
part of the problem right there. But we won't get into that because that's another issue. But yeah, so the de novo case, you just have to be carefully examined so that but if there's a mutation and they meet HHT, that they have that mutation, then they'll pass it on. They're just the first. The mosaic is different because the mosaic says that the mutation didn't happen in the sperm or the egg, but in the developing embryo of one of the parents. So they're not every cell in their body is the mosaic. So the question is this: what about their germline? For the
Mhm.
male, what about the cells that eventually become sperm cells, the spermatogonia or whatever, that are developing into the sperm? Maybe they don't have all of the cells have a mutation. So the frequency passing it on to the next generation might not be 50-50. The problem is predicting mosaicism in the germline from mosaicism in the soma, in the rest of the body. So,
That's
but
really
yes, invariably it probably is that the germline is the mosaic and the frequency is going to be less than 50-50. But
right.
predicting what it is, good luck.
Yeah, that's always confounded me. If someone has an HHT diagnosis but doesn't know their specific gene mutation, should they consider seeing a geneticist, and what would be the benefits?
Yeah, that's an interesting question because presumably that means that they did have testing and it was either a VUS or
Well,
just zero, like they got no
or
result
they
back.
may have just a clinical diagnosis and never
Yeah.
got genetic testing.
Yeah, I still think that the geneticists and the genetic counselors are very important as
Mhm.
a team. The geneticist isn't going to do anything, but like if you go to a center of excellence, I think you need to think about the fact that the geneticist understands the history and the inheritance pattern and some of the issues. That the pulmonologist is looking at the lung and the neurologist is looking at the brain. The geneticist is saying looking at the family. So I think it is valuable to see a geneticist. There's one more point that I want to make that I probably shouldn't, but diagnostic labs are very risk averse. So they are very careful. And if it's like, I don't know if that's a mutation, I better call it a VUS.
Mm,
I'm not a medical geneticist, but I've seen lots of people email me all the time and say this was my VUS, and I do my own analysis, and I say, well, I can't be sure either, but boy, oh boy, that to me looks like a mutation. And just if you don't think that just happened this morning, and somebody wants me to go later on and meet with
wow.
the doctors and here at Duke, and they say, well, the patient had a VUS, and they want patient has HHT, there's no doubt about it. And I said, send me that genetic data. I want to do my analysis because
Interesting.
I might give a different opinion, not because I use any different tools, but I'm not putting my neck out on the line and giving a clinical
Right.
diagnosis that's going down in the medical records and is being paid for. I'm just going to say my view is that's possibly a mutation, So
What
that
would
a
you
geneticist might be willing to do and not necessarily as a clinical report that gets put down.
what would you advise families, especially, and I'm sure you may have come across this, um, in pursuing genetic testing when there's fear or hesitation?
Yeah.
That's a tough one.
That is a tough one. And people are, number one, are always worried about insurance and
Discrimination.
discrimination there. Although we have the GINA Act, the Genetic
Right,
Non-Discrimination Act that was passed years and years ago,
right.
there's still the fear. And I kind of understand that because then they change jobs and it's a new employer, a new thing that's now pre-existing. I think that the value of the genetic test is if you have children or relatives who really need to know what's going on. So for me, I think that is really important, especially for the kids,
Yeah,
because you don't want to take all your kids and like be looking at them and saying, "Gosh, I don't know,"
right,
as they're growing up. You really want to focus your attention on those that have it, and not to discriminate or anything, but just to say we really need to be watching Junior very closely and not,
right.
Mary or whatever. Yeah.
Yeah, for sure. I know we have some resources on our website about that too. So, are there any automated or standardized tools available to help analyze HHT genetic variants?
Yeah, so we've talked. I have a whole series of slides when I teach class to the graduate students about some of these tools, SIFT and Polyfen and CAD and others. And so I don't know if I'd call it AI, but it is computer based, really trying to make predictions. So they're very, very good, but they're, like I said, they're only predictions. One thing that people do use is instead of using one of these algorithms or two, they'll use a whole suite of them. They'll basically throw the entire computer system on on the variant and say, gosh, if they most of them agree that it's a mutation, maybe we should elevate that to becoming a mutation. I want to say one thing more, and I think I saw that Anthony Anzel is here, who should be giving this talk as well. He did some beautiful work at going back into these big population databases that I talked about and saying, hey, these are all supposedly quote unquote just random, normal, whatever that means, people. And he found lots of people using very sophisticated algorithms that probably have HHT. And so the incidence is probably even higher than what people think because he just went looking in the sequence. So he used a lot of these tools and he had a very sophisticated way where he had like different tiers of confidence. If you want to be super conservative, or super aggressive and say we're going to call anything that we think, and so I mean it's a little bit of a difficult paper for the non
Yeah.
scientists, but yeah. So
That was,
Anthony,
yeah,
hello,
that
you're
was
there
really
still.
good. I mean, that really went right into my next question, which was about using genealogy or population genetics. So you
Yeah,
answered ahead of time. So we
yeah, good.
appreciate that very much. I'm going to turn it over to Cassie.
Yes, I have always so many questions for you, Doug. I could pick your brain forever. Okay, so going back to genetic testing and this idea of having a variant of unknown significance whenever you get your result, but you are having clinical symptoms, your family has been clinically diagnosed with HHT. What happens later? I've heard of people
Okay.
receiving letters from the genetic testing company saying that their gene status has been updated. Does that mean anything?
Yeah, thank
What
you. That
happens
is a
next?
and that was an excellent question that something I didn't cover. When you get your test, it's a moment in time. 2 years from now, there's going to be many more mutations found, and they're going to see, oh my gosh, four different independent families had that thing that we thought was a VUS. And in fact, there may be other evidence from splicing mutations or new algorithms that they could rerun on that variant to say, the splicing algorithms are getting better and better because splicing is one of the hardest ones. So, And the snapshot of time you got the result and it was a VUS, and 2 or 3 years later, they would have called it a mutation. So I think they have an obligation to get back to you and say, whoops,
Mhm.
not whoops, but we think we've reclassified this as a mutation. And it will always get better.
Yeah. I
Yeah.
think that's a good incentive for people to get genetic testing, even if
Yeah,
they already have a clinical diagnosis, because you're adding to that knowledge base,
that
you
is a
know,
good
that
point.
will help people over time.
It will help others. It will help the CCM HHT community, even if it doesn't help you right away.
Right. And so how do you mentioned that there are like computer algorithms that can look at specific mutations or, missense frame shift and kind of predict what it might do to the protein. Can you do that in models in the lab? Like, how does that happen? And, is there a certain variant that's maybe more prominent in the population that people choose to build into cell models or animal models of the disease?
Yeah. I think I think if you were going to try to make an animal model or a cell model, you wouldn't necessarily do that on a variant that you weren't sure was a mutation because that's a huge amount of work. But this issue of doing sort of standard cell molecular biology by looking at the RNAs that are made, looking at longer reads. So yeah, but to make an organoid, for example, or something, you would make it. You could make it on that variant and see if. But I think that's really labor and time and personnel expensive. So the people who've made organoids and cell models and animal models, they've made it on mutations that they're rock solid sure because they want to study
Mhm.
the downstream pathobiology. But you could test that and do a functional test. The person who I really admire a lot is Sabine Bailey from
Mhm.
Grenoble, France, who many of you know. Sabine has taken a lot of these missense changes, and but boy, this is a lot of work. She's put them
Yeah.
into cells, and the cells make the protein, and she studies the signaling, and then they say these are mutations and these aren't. But
Mhm.
boy, that's not going to scale well.
Yeah.
Beautiful work, and I love her work.
But you think that, because that isn't very scalable and most HHT families have different variants that, maybe AI or the use of these computer algorithms are going to play a bigger role in personalized medicine for the
Yeah,
disease?
yeah. I think AI and these computer algorithms are going to help, but I always have a caveat here, and this is my bias. AI only works if you have a lot of data to give it so that
Yeah.
it can do. So we need everybody to be sequenced then and put all that data in and put all the clinical information in, and then AI will help. And I do think also this if we're going to start learning rules about regulatory, how the genes turned on and off, we don't necessarily only get that information from HHT, right? So
Mhm.
as all these diseases are being studied, that's where we'll have this huge database. What are the variants that can alter the expression of the gene that don't have anything to do with the sequence itself, the
Mhm.
protein sequence? That's going to come not only from HHT, but from lots of diseases, lots of data, and that's where AI is going to have to be. It's just the AI is, I think, really good when you've got a data overload.
Mhm.
I don't know that we're at data overload for HHT alone yet, but
Yeah, we're
but it'd
working
be
on it.
nice if we were.
I
But what
know.
we can do is use everybody. That's why I says there are lessons to be learned in human genetics from all the other studies that we
Mhm.
can use on HHT. So we shouldn't think that HHT is that different.
Okay, so one more question because we have, I think, quite a few questions submitted from
Yeah.
our participants. Do you think because of the variants or because of maybe genetic modifiers that change the expression of genes, do you anticipate that once therapies are out and about for this disease finally, that different people with different variants will respond differently to
Yeah.
certain therapeutics?
Well, I mean, people always respond differently for different therapeutics for many reasons that don't have
Mhm.
to do with the mutation. So my own view is, and I think this is right, that that there may be some therapies that only work with endoglin patients and some that were out, but we only have three genes, and one of them is very unique with the SMAD4 with the JPHHT. So if you think about it for garden, I don't, that's not the right word, but the genetic garden variety HHT that's HHT alone, there's only two genes. So I don't think that the therapy is going to be mutation specific. I don't even think it always has to be gene specific, but even if it's gene specific, we've only got two genes for the that kind of HHT. So I don't think it's going to be mutation specific, and I don't think people should, oh, I've got a weird mutation. If it
Yeah.
messes up endoglin, it messes up endoglin. We may not be able to figure out how it messes up endoglin, but it messes up endoglin. And
Mhm.
a therapy that is for endoglin patients should work. Will it work equally on everybody in the same way that any medication you take is different and people respond to it differently? That's a separate topic, but it's not on the basis of the mutation itself.
Excellent. All right, Helen, back to you with more Q&A.
Perfect. Thank you so much. We're going to go ahead and move on to some of the questions that have been submitted through the Q&A and through the chat. Um, and Cassie and Marianne, please feel free to also answer and dig in with us. But the first one is, if a patient is DX with SMAD4 and seems to be the only one in the family for generations, what genes should the family member be tested for?
Wait, if they're diagnosed with,
SMAD4.
so SPAD4 is the gene. So if they've already got, if they've been shown to have a SPAD4 mutation, then that, I think they've got SPAD4. They should test everybody with SPAD4. If the question was if they have JPHHT, they should sequence SMAD4. I think they would sequence all three genes, but it'd be SMAD4. I think the way the question is worded, I'm not sure whether they're worried about the diagnosis or the genetic test.
So I
But if
think
you have the phenotype of JPHHT, you should, I think the diagnostic lab would sequence SMAD4. They probably sequence all three genes, but you would sequence SMAD4 first. This issue of who the diagnostic labs have their own algorithms for how broad or like I know the Utah group has a panel of a bunch of genes, and they throw everything on there because it's actually anything with vascular malformations because it's more cost effective to do it in a pipeline like that than to be doing one-offs for everybody. But SPAD4, if anybody has diagnosed with polyps and HHT and they think, these juvenile polyps are unique, they should be tested for JPHHT with a SPAD4 mutation, yes.
And like Doug said, with the first person in a family to be genetically
Yeah,
tested for HHT, they typically do the full panel, like a vascular anomalies. They'll test all the potential HHT causing genes. But then once that genetic mutation is found, so if it's in
yeah,
SMAD4, then anyone else in the family just needs to be tested for that gene because
yeah.
that's the one that's inherited.
Yeah, so that's the knowledge that once you have somebody above, then you could test your kids. You don't sequence all the genes, right? So I didn't make that clear, but yeah, it also reduces the costs tremendously. I don't know actually what they charge, but in principle, it's trivial to do that because you're only looking for one mutation.
And speaking a little bit more about testing, how beneficial is it to pursue testing beyond the gene? And for example, this person said that all I know is that our family has the ALK1 gene, but that was 25 years ago.
And so how beneficial is it to,
To pursue testing beyond the gene.
I mean, I guess I my worry is when they said 25, was it 25 years ago?
25
Just,
years ago.
yeah, I think it'd be important to go to a genetic counselor and say, that was 25 years ago. Let's look at that test result and see who did it, what was done, because I don't know. But if as Cassie pointed out, if the genetic counselor looks at it and says, this is a bona fide ACVRL mutation, ACVRL1 mutation, then then continue to test for that in the family. Yeah.
Is it prudent for an individual with HHT and contemplating marriage to have his or her intended spouse to take a gene test to see if the possibility exists that both may be positive for HHT?
I know I saw that I was looking at the questions in the sidebar here and I saw that one. I thought, okay, what am I going to say about this one? Yeah, I would think that if the person knows they have HHT and they've been tested and they know their mutation, and presumably they're getting married to a person of approximately equivalent age, and that person shows no signs of HHT, no family history of HHT. I mean, you could sequence, but I don't know that would be. I would think it would be unfruitful. And by the way, what would happen if two people had HHT and they had a kid who got mutant copies from both parents? And the animal model data says that that's not a viable fetus in all sorts of mice, mouse models, and others. And if I had time, I would talk about some of the old papers that had conflicting results with the human. But the point is that if the person doesn't have HHT, doesn't have a family history of HHT, and they're in marriage age, it'd be interesting. What would you do, Cassie? I mean, you could have the sequencing done, but there's no reason to do the sequencing, in my view.
Yeah, I would say there's probably not a reason to do it. And even if you know, every family has a different variant, and so that's
Yeah.
also probably in consideration. You know, you probably, if you're marrying someone who did have a family history of HHT and they got genetic testing, may not be the same gene or the same gene
Yeah,
variant, so it may not have the same impact on something, you know, like the viability of a pregnancy.
yeah. I think the thing is that you don't have to have a clinical. This is the key: is the
Mhm.
testing is not gonna could tell you you are at risk of developing these symptoms because you have a mutation. But if a person isn't showing symptoms by a certain age and they have no family history in the past,
Mm. So is HHT, I've heard it called an age-related penetrance kind of
yeah,
disease.
okay.
So
Right, because
what does
because
that mean?
over time, we start to see the development. Were the lesions always there or not? In some cases, it's definitely not because the ones you can see on the face and the skin and everything, they seem to appear with age. So age-dependent penetrance and expressivity means that you can maybe start more epistaxis in later age. And age-dependent penetrance means that you can't tell whether the person is HHT possibly until a certain age. I think some people would say that, well, if you get a mutation, you don't necessarily have HHT. And I'm not a clinician, but I would think that by a certain age, maybe in the 40s, if you have a mutation and you're seen at one of the centers of excellence where they know what they're looking for, I think they could make a pretty good diagnosis. So there's a penetrance is going, a young child may not show the symptoms yet, or just show a little bit of nosebleeds. We're not sure. We don't see any telangiectasia. So it does take time to develop over time to make that diagnosis because the symptoms come on over the decades and seem to maybe get more severe over the decades as well.
The question submitted was, am I understanding correctly that we don't truly understand the functions of each gene, or is it that we don't understand the specific effects of the mutation within that specific gene?
Well, okay, so let's take the latter part first. That's at the level of the gene to the protein. And for a lot, except for the ones that we can't like the VUSs, we know very well that these are what are called loss of function and they break them. So that's clear. So when you have a mutation, it messes up endocrine function. And we know what endocrine does pretty well. And we can study that biochemical pathway, and we know that when you have the mutant version, that pathway is perturbed. How does that biochemical pathway in the cell, in the body, create a vascular malformation? And that actually, there's some interesting data coming out on that, but that's the hard part to understand how that mutant cell, those mutant cells eventually develop into a vascular malformation. But we understand how the protein works. And that's the basis of some of these newer, like diagonal therapeutics, of attacking the actual pathway that's mutant. And that's, I think, among the most exciting things going on in HHT right now, is attacking where we know because we know so much about how it works. We just don't know how that ends up being a vascular malformation. The vascular malformation is very anatomically interesting and different and hard to figure out how did you go from a normal vessel to that. And even though you can say you could draw a picture of what's happening, how does that biochemistry create that?
Okay. Thank
Yeah.
you. And I know a little bit earlier, correct me if I'm wrong, you had mentioned that it was a loss of function, but why is it assumed that HHT is a loss of function disease?
Yeah, because all, here's yeah, the spectrum. Now I'm giving my graduate human genetics class. The spectrum of mutations can tell you the mechanism of the mutations even before you do any biochemistry. So you can machine gun that gene and they all cause HHT. So think of like your car. If you give me a hammer, I'm going to screw up that car if I start banging on the engine. Will I one day bang on the engine and make it go faster? Possibly. But the thing is that all those mutations are consistent with loss, like the stop codons. The stop codons that don't even make a protein are almost nothing. How about the deletions? What is happening when you don't even have the gene? Well, that's a loss of function because it's a loss of the whole gene. So the spectrum of mutations across all the patients shows that it's loss of function. And that's the conundrum. How do we fix that? Maybe the best way is just to add it back
Hmm, interesting.
and not try to correct it, but just throw it back in.
Yeah.
Now you got another copy.
Thank you. And then we'll go ahead and end with this question, and Cassie and Marianne, please feel free to also add to it. But what does the latest science say about the prevalence of HHT in a population?
Yeah.
I know we've heard 1 in 5,000. We've also heard 1 in 4,000. So can you talk a little bit more about that?
Yeah, I'm trying to remember the details of Anthony's beautiful paper because if you tried to do it from centers, well, then, and say, well, how many people live in, North Carolina and how many came to a center? And you're trying to estimate the prevalence based on clinical phenotype. That's going to be hard because not everybody comes in. And like we talked about before, so many people weren't diagnosed for years and years. So what Anthony tried to do is go into these big databases, and he found that the prevalence is way higher
Mhm.
than what it is. And I can't remember the exact numbers, but again, it depended on how. Here he is. He wrote. I gotta open this up.
It suggests 2 to 12 in 5,000 at the
Well,
most.
okay, so yeah, 12 in 5,000, 1 in 500, that's right. So that's a lot more prevalent. And again, it depends on how aggressive you want to call that. That's why these prevalence estimates of diseases that aren't so obvious right from the get-go, where they can do population studies. So I think I think Anthony's paper is one of the most important papers to come out, and we saw it in its poster form at the conference in France, and I thought, wow, this is going to be an important paper, and it is. So it's much more prevalent, and if he's right, it's like maybe 1 in 5,000. And there's some clapping going on.
So
So
so that's really interesting, and I think a couple factors too I want to put out there. We just talked about this yesterday, actually, during our congressional briefing about prevalence, and basically, we have a long diagnostic odyssey.
yeah,
And so it's anywhere between, our latest paper that was just published. I'm going to put a shout out for the chorus paper that was just published yesterday in Blood. It's huge. It's the first time we actually have real data from our registry, but it's like 21 years.
yeah,
And so you have to think that way, that we have this long diagnostic odyssey, but on top of that, um, it's every time we are diagnosing more people. When I first started, Doug, and you and I began our work together all these years ago,
yeah, yeah,
we always said 90% are undiagnosed. Well, we've moved the needle, hopefully, all these years. We are increasing diagnosis rates, but still, it could be anywhere between 60% still undiagnosed. And so when you think about it that way, you know, the more people you diagnose, the more prevalent your disease is becoming, right?
yeah,
And so I personally think it's much more prevalent, and
yeah.
we just need the data. Cassie, you have any comment?
Yeah, I pretty much agree. I think the somatic mutation story we didn't really get into. I think, Doug, that
Yeah,
could
yeah,
be a
yeah.
whole second webinar. But because of that, that's primarily what causes the high variability
Yeah,
between
yeah,
patients
That's
You
kind
know,
of why I wanted to talk about it, but it was too
yeah.
much.
It's really hard to wrap your head around too. And but that kind of genetic part of the story is is really telling for, you know, who is going to experience symptoms and how severe those symptoms might be and how variable those symptoms are. So you may have someone with an inherited HHT mutation, but not really have any internal organ AVMs, not very symptomatic for their whole lives. I'm sure many of us have those people in our families that are like
Yeah,
that. And so they're probably never counted in the prevalence rates because
yeah,
they're not seeking care or a
right.
diagnosis.
This
And
was, yeah. Sorry. Good.
a
I'm
lot of
sorry.
people just don't know either. They're
Yeah.
walking around and they just don't know.
Can I address one of the questions that I spent some time doing and we didn't come up with this? Someone asked about like, are the platelets dysfunctional in HHT? And endoglin and ALK aren't expressed on the platelets. And when people have pulled the platelets out of people's blood and done this, really careful analysis of the platelets sitting in a test tube and running it through all sorts of tests in a machine, their variability is no different than the normal. But the platelets in the body are influenced by the endothelial cells in the blood. And Carmelo Bernabeu had, gosh, 20, 15, 20 years ago, showed that there are defects in the endothelial communication with the platelets. So even if the platelets are normal, they have to function by interacting with integrins and all sorts of stuff, including the endothelial cell layer. And endoglin and ALK are sitting there right on the surface. And I said again, Carmelo Bernabeu's some beautiful work years ago, and still he just had a review a couple years ago, really reviewing the parts of the endoglin story, especially endoglin, that are related to platelets. That not that the platelets are bad, but that the interaction between the two, the cells, the endothelial cells and the platelets. I wanted to do that because and the reason why I have my notes, the reason I wanted to say this is because the hematologists like Raj Kasturi and these guys, they're really active in the HHT community because we would love to stop the bleeding or curtail that as best as possible. And there may be some role for there is definitely a role for studying that and thinking of new therapies, not to try to reverse the lesion, the vascular malformation, but just to stop the bleeding because there is this defect in the communication. That's why I want to say it because it has a therapeutic implication. There were,
Thank
yeah,
you. Yes.
I'll stop.
I know I'm like this is a conversation that can just go on, go on, go on, and
Yeah,
we can keep
I
learning
wanted
more.
to handle that one because I think
Yeah,
I want to do shout out to some of the people who for 20,
absolutely.
30 years have been doing beautiful work. And Anthony's more on the front end of that with some of the newer stuff that he'll be doing going on in the future. He could come here to Duke and take over my lab, but
Maybe,
I hope,
maybe we'll do another webinar on somatic mutations. I think
yeah,
that's going
maybe
to be
someday. Yeah,
like round
I would
two.
love to do that.
It's an
I
important subject all on its own because you've really cracked why different family members are affected differently with this disease, which always confounded
yeah,
everyone. And so, Doug, you've made that discovery, which is just so dramatic for
Absolutely.
our population.
and now it's time to
Absolutely.
leave it in the good hands of the new generation of really smart people who we're going to get it. I am convinced. I don't know about the gene therapy because I think that takes a little longer, but we will have some really good therapies very soon. And then I think gene replacement could be an option. Not correction, but just add back. That's so easy. Okay, anyway, good. All right,
Well,
thanks
thank you.
everyone for listening.
Absolutely. Thank you so much, Dr. Marchuk. Again, what an amazing presentation. I also want to thank Cassie and Marianne for moderating today's webinar. And on behalf of everyone at Cure HHT, I do really want to say thank you to everyone attending today's webinar and for staying connected with us. We really do hope you enjoyed it. And again, thank you again to Dr. Marchuk. Dr. Marchuk, you have a few comments in the chat just praising you and just wishing you well in your retirement. And thank you really for sharing your expertise with us today. And as I mentioned in the beginning, this webinar is being recorded, and you will be receiving a link to access it on demand next week. And if you missed any of our previous webinars this year, we do encourage you all to visit our website and watch them at your convenience. And then one last thing I'll ask from everyone is to please take a moment to complete the survey that will also be sent to you via email. Your feedback is incredibly valuable and will also help us improve future programs. So we truly appreciate your participation, and we look forward to seeing you at our next webinar. Thank you again, Dr. Marchuk. Thank you, Cassie. Thank you, Mary Ann.
Bye-bye, everybody.
Bye, everyone. Have a great day.